Two-photon excitation microscopy, also referred to as multiphoton microscopy, is a type of fluorescence microscopy technique commonly used to image living tissue. Fluorescence microscopy is a type of light microscopy in which fluorescent molecules in the sample are excited by high-energy photons. After being excited, the fluorescent molecules emit longer wavelengths of light, which are then captured by a detector. A light source is passed through an excitation filter to select a certain wavelength of light. Light is reflected off of a dichroic mirror, which reflects or transmits different wavelengths of light differently. Emitted light from the sample is then captured by photomultiplier tubes after passing through an emission filter. However, in samples over 2 micrometers thick, there is a noticeable out-of-focus flare due to secondary fluorescence emitted from regions of the specimen that aren't of interest. This interferes with the resolution of features that are in focus. Confocal microscopy uses a focused laser beam as the light source and adds a pinhole in front of the detector to address this issue. The pinhole blocks out-of-focus light during image formation to produce a clear image. However, confocal microscopy still uses high-energy photons, which lead to phototoxicity and photobleaching and ultimately reduce the viability of living tissues. Additionally, penetration depth is limited by light absorption and scattering in samples. TPM can improve the detection of signal photons and penetration depth even in highly scattering environments while minimizing photo damage to the sample. Two-photon absorption is when two photons interact with the same molecule at the same time to excite a fluorescent molecule the same way a single high-energy photon does. Same time means within a time interval less than 10 to the negative 18 seconds. Using longer wavelengths of light, specifically infrared light, is beneficial because many biological molecules have much lower absorption of light at those wavelengths. In TPM, a pulsed output laser, typically a titanium-doped sapphire laser, is used. When two photons simultaneously interact with a fluorescent molecule and excite it, a photon is emitted from the sample. The emitted photon then travels to the PMTs. Since two-photon excitation depends on simultaneous absorption, the fluorescence emission varies with the square of the excitation intensity. This means TPM requires a higher intensity and a more powerful laser than confocal microscopy. A pulsed output laser is used instead of a continuous wave laser to achieve the necessary peak power without exposing the sample to a damaging high intensity for long periods of time. Even though the laser is scanned over the entire specimen, fluorescent excitation only occurs when the two photons interact with a molecule at the same time. This means TPM has a much smaller focal volume than the focal volume seen in confocal microscopy. It is important for the photochemical interaction volume to be small when imaging biological specimens in order to minimize damage to the specimens. Focal volumes can be as small as 0.1 micrometers in TPM. That's nearly 1,000 times smaller than the thickness of a human hair. TPM is used to image biological specimens, which must be prepared in a specific manner. The preparation of a sample can be broken down into 3 steps: fixation, permeabilization, and staining. Tissue fixation helps preserve cellular organization. Permeabilization is necessary to allow staining solutions to pass through the plasma membrane and access internal cellular structures. The staining of a sample, done by using a fluorescently labeled antibody, makes the sample fluorescent and gives it visibility in the microscope. TPM can be used to characterize cell structure of living sedated species, typically rats, or more commonly prepared cell specimens on slides. Under optimal conditions, TPM can image up to several hundred micrometers in depth, typically 6-fold deeper than confocal microscopy. While TPM is a very useful tool for imaging biological specimens, it has some limitations. If the tissues in the specimen are densely packed, the fluorescent signal will be weaker and it will be difficult to image deeper than 300 micrometers. TPM also has a lower resolution than single photon microscopy, and there is potential for thermal damage of the specimen. Additionally, using a laser as the source can be very expensive. I hope you guys enjoyed, and thanks for watching.